I'm Alex. I'm the CEO and co-founder of LightSwitch Bio. We're a small pharmaceutical company working to help patients avoid the side effects of chemotherapy using a new method of drug delivery that is light activated. So I can't get my screen to, there we go. So everybody may be familiar with the side effects of chemotherapy. They're just generally awful. And one of the best examples of that is from a drug called cisplatin. It's used to treat about half of all patients who receive chemotherapy, and pretty much everybody who takes it gets one or all of these side effects. And the cost, the added cost for patient care to deal with these side effects, from follow-up monitoring or just new medicines to deal with, like the nausea, for example, can add tens of thousands of dollars per patient. And about 40% of the patients who take this drug end up having toxicities that are so severe they have to stop care and move on to another form of treatment, which is generally also more expensive. So our solution to deal with these side effects is to take cisplatin and then bind what we call a photocage to it that makes it inactive in the body, and allows us to shine a laser on the tumor and allow selective drug release just there. This photocage acts like an antenna that absorbs the light energy and breaks that bond. It also allows it to be fluorescent, but I'm focusing on the release for now. And while it releases the drug, it also releases cytotoxic single oxygen, and these two mechanisms are known to be synergistic in a wide array of cancers. And we've just been awarded a patent for this in the US. We're currently still prosecuting for EU protection, but this patent is owned by VCU, and we've had an exclusive option of it for the past few years. Because cisplatin and platinum therapies and the cytotoxic singlet oxygen mechanisms are so applicable to a wider range of cancers, we think we could potentially treat all of these different types of indications. However, due to patient numbers, accessibility of tumors for a laser source, and the need for new therapies, we're initially focusing on head and neck cancers, and then we'll move on to mesothelioma and lung cancers and further down the line. So we initially selected head and neck cancer because it's incredibly common, it's incredibly deadly, it's the costliest cancer to treat, and the morbidities are very severe. It's the treatment options tend to cause a lot of mouth inflammation, which makes it difficult to eat. They can also cause a lot of facial disfigurement, which nobody really wants. And regardless of the subtype of cancer or the stage of cancer, cisplatin is built into the standard of care. So we believe that if we can help patients avoid those toxicities and allow them to actually receive cisplatin treatment in their tumor, we can improve care for many of these subgroups, if not all of them. We formed a team to help develop this therapy, comprised of myself and Dr. Hartman, who's the inventor. And we fleshed out our board with clinical oncologist, Dr. Popovic. He's also the medical director of the clinical trials office at Massey Cancer Center. So he helps us strategize on the clinical side of the development path. And then we also work a lot with Joff Masakawa, who's worked in senior leadership roles in several pharmaceutical companies, and currently runs a consultancy focused on commercialization strategies for specialty biopharmaceuticals. And together, mostly from the rest of my team, we have a combined 65 years or more of experience in developing novel therapeutics and oncology indications. We further fleshed out our team with a suite of scientific collaborators at Massey Cancer Center and University of Pennsylvania. So Dr. Harada and Koblinski are experts in mouse models of head and neck cancer. And then Dr. Bush and Zhu are experts in phototherapy, development, and Dr. Basu helps really refine our clinical approach for head and neck cancers. We've also received additional support by working through the Mass Challenge Accelerator program. And we've also received some funding, which we've been very fortunate. So for the past few years, we've operated off of about $75,000 in commercialization grants and pitch awards to help earn that IP and generate some pilot data, which we've then leveraged to earn a phase 1 STTR award that's going to help us develop the therapy more in mouse models and help us get to a pre-IND meeting with the FDA. So this is some of the pilot data that we've generated with IR-Platin, the light activated version of cisplatin. So on the left, we're looking at the potential safety of IR-Platin, and we're looking at the weight change in mice after they've been delivered the drug. And then on the right, we're looking at an indicator of efficacy. So this is measuring growth of the tumor after treatment. And so when there's no treatment, the green line, the tumors keep growing. And so you see the line drop off to zero because all the mice are no longer being considered surviving. So that's an endpoint indicator. And then when we treat with cisplatin, tumors generally still keep growing, except the tumor stops growing for a while and then it continues to grow beyond that initial size. But we see that when we treat with just 5% of that equimolar dose with IR-Platin, the tumors stop growing and they start shrinking. And in about a third of the mice that we treat, the tumors go away completely. And we waited 90 plus days and the tumors never regrew. And that's to us indicating a anti-tumor immune effect. And that could be due to a combination of the mechanisms of the cytotoxic singlet oxygen and the cisplatin, and causing immunogenic cell death, or it could just be because we're avoiding the immunosuppression that cisplatin toxicity causes. So we're really excited to see that, but unfortunately, we're not able to assess that with the $400,000 STTR award that we were given. That's something that the reviewers were a little disappointed about, that they can only award so much funding. So that's what we're currently focusing our current raise on. So in terms of market competition, we're looking at other platinum therapies and phototherapies as our closest competitors. And both of these are really dominated by 3 off-patent generics. And we hope to really benefit or really combine the benefits of both approaches and hopefully avoid some of the downsides of both with IR-Platin. And when we look at what we might be able to treat with IR-Platin, we want to be able to treat those indications that are treatable by both of these. So something that is treatable by platinum and accessible with a phototherapy. And when we do that, we get up to a potential of 1.5 billion current market value, and that's growing over time for about 3 million patients. And when we narrow that down further to locally advanced and recurrent head and neck cancers, that gives us about 200,000 patients that we could potentially treat today. There are other potential therapies that we consider competitors. I mean, really every therapy that could be used to treat cancer is a potential competitor, but aside from platinum therapies and phototherapies, we think the closest are other biologics. So immunotherapies and then photoimmunotherapies, where they take one of those biologics and apply not a photocage, but a light activated molecule that helps either increase the toxicity or aggregate those molecules and cause the cells to lyse. However, those protein-based therapies are generally very expensive to make, and we're using a small molecule approach that is cheaper. So we can help patients hopefully avoid some of the financial toxicity that causes them to avoid cancer treatments or even going to the doctor to see if they have cancer. And so, like I mentioned, the SCTR award that we were just awarded about a month ago, that's going to help us further evaluate IR platinum's cytotoxic efficacy, its toxicity in mice, and some of its pharmacokinetics, but it's not going to let us evaluate that potential for an anti-tumor immune effect or evaluate how it works in larger tumors, because the size of the tumor is going to be a limiting factor on how deep can we treat with light. And part of the contributing factor to the rapid growth of other phototherapies is due to better methods of light delivery that allow larger and deeper tumors to be treated. But until we do those studies, we can't say for sure if it can treat human-sized tumors, like the full scope of human-sized tumors, because we're currently in mice. So we're currently trying to raise another $500,000 to flesh out our development with those anti-tumor immune studies. And that will take us to a pre-IND meeting where we will figure out what exactly we need to do with GMP manufactured compound to reach human trials. And then that's going to take us another 7 to 14 years and $45 million to reach market. And if we can reach market and deliver drug to patients with a cost of goods of about $2,500 and sell it at $5,000 and still save patients $10,000 by avoiding side effects, we should only need to penetrate 9% of the head and neck cancer market in order to break even on all of our research expenses. And so this is really just our financials, and we're going to have to decide when we reach human trials, do we want to sell or out-license or continue developing all the way through human trials? I think we can go all the way to human trials or through human trials. But that's the end of my presentation. I simplified a lot and I glossed over a lot just to try and keep things short, but I'd love to answer some questions and connect further offline.